Ni hao, Internet. This is your captain speaking, Lester Nare, joined as always by my co-host, our resident PhD, and number one Chamath Palihapitiya cosplayer, Krishna Choudhary, my friend.
That's right. I can't afford the sweaters that he wears.
The alpaca shipped from God knows where.
Yeah. This is nylon shipped from TikTok shop.
You don't have the SPAC money.
No. Maybe one day.
One day. Maybe not via SPAC, but via science communication.
Yeah.
It is now, we are in the holiday season. So we are winding down for the year. Big shout out to everyone who's joined the show. We started less than 6 months ago.
Yeah.
The response has been incredible. We're going to be wrapping up season 1 basically at the end of the year. I think we've made the random executive decision to decide our seasons are just going to be every year.
Yeah.
And so we started only in halfway through this year. So season 1, a little truncated.
Yeah, no,
Pilot
it's good.
Season.
No, the Gregorian calendar is a great calendar.
It's a great calendar. So that's how we're going to do our seasons.
Yeah.
So this is the first of kind of our wrap up of the year episodes.
Yes.
With holidays, with travel, all these things, with Mariah Carey being on the radio nonstop.
Yeah. She's creeping up.
It's the holiday season. So what we're going to do this episode is similar to what we've done when we did the Nobel Prizes.
Yes.
Which is we're going to do a single story, deep dive. And what we're going to talk about in this episode is the discovery of DNA.
Yes.
And the reason why we're going to talk about the discovery of DNA is the unfortunate recent passing of one of the pioneers of the discovery of the double helix, which was James Watson, who recently passed away.
Yes.
And there's a lot of drama in this story. This will be a complex episode. We are really focused on science, experimental design, the concepts, thinking about things from first principles. But this is one of the things I think we both do is it's a human story.
Yes. And it's a fascinating human story.
And there's a lot of controversy associated with Mr. Watson. And so we're not going to shy away from...
No, not at all.
Talking about that in the context of what was one of the greatest discoveries
of the 20th century.
Yeah.
Easy.
And as always, you guys know we're going to go deep. It's going to be fun. It's going to be lighthearted, but serious. You're going to learn something today. This is From First Principles.
So, James Watson.
Yes.
He recently passed away, and colleagues and people in the field are wrestling with his legacy because he is a very controversial character. Okay? He is clearly one of the greatest scientists of the 20th century, right? Discovering the structure of DNA is an incredible achievement. It is quite possibly, I would say, in the world of biomolecular chemistry, it is the crown jewel of human achievement up till now, is the discovery that DNA is this periodic, double helix, twisted ladder, beautiful looking molecule that is ubiquitous in all of life, from bacteria, archaea, fungi, plants, animals, all the way to us. It's something that tells us that we're all connected. Right? Me and the plant and the bacteria, we all have the same lineage. One of the definitive proofs of that is the fact that all of us use DNA as our information molecule, right?
It's the idea of this universal storage system.
Yeah.
Like everything that we classify as life uses the same Samsung hard drive.
Yeah. Everyone uses like the same. Yeah, it's like a transistor. In some sense,
fair.
it's everywhere.
It's everywhere,
and it's so good at what it does. And one of the reasons why it's so good at what it does has to do with its structure. At the end of the day, biology, I've told you, is just lock and key, right?
Right.
And the shape of these molecules has everything to do with what the thing is. And DNA is one of these things that's just incredibly beautiful to look at, incredibly beautiful to think about, beautiful to innovate on as a technology. At the same time, James Watson is a very controversial figure. He helped strengthen U.S. research. Like he single-handedly did a lot for U.S. biomedical research, but at the same time, he was very controversial. He said some really out-of-pocket things towards the later years of his life.
Sorry, I'm looking for my pocket, but it's kind of out right now.
Yeah. Yeah, dude, later on in the story, we're going to get into some of the stuff he said, and they're like comical how bad they are as takes, right?
Look, as a member of the Black community, it's exactly like the story of R. Kelly.
Yeah.
Great music.
But man. You really?
You can't. It's tough.
Yeah, it's tough. It's tough. But you got to celebrate the good with the bad, right? And you got to know all of the nuances of the story to really appreciate it.
It's the human experience.
Yes,
right?
It is. Yeah. And I want to start by just talking about the historical landscape that sets up DNA as this molecule that everybody is going after. Mm-hmm. Why it was such a big deal that we got this structure. And then we'll go into some of the future of, after the 1950s, when this was discovered, what happened to the players. Okay?
Okay.
So we're going to start actually all the way back in the 1800s with a man named Gregor Mendel. Mm-hmm. Back then, everyone knows about like inheritance, right? Like you get stuff from your parents, but nobody really knew how. Gregor Mendel is this guy. He's an Austrian monk, and he does experiments on pea plants. And he starts noticing that actually when you do these quantitative experiments, you can really mathematically say how much of the next generation is going to get a certain trait from their parents. He was doing very selective breeding where, for example, let's say a pea pod had green and then there was another pea plant that had yellow pods, right? If you selectively breed them together, how many of the next generation are going to inherit the yellow versus the green? How many of the next generation are going to inherit tall versus short? How many are going to do this or that, right? And there was a mathematics to it. There was an understanding that you could glean, which means that it
wasn't random.
It wasn't completely random.
Right.
There's some mechanism that's happening, and this is before the whole mechanistic view about biology.
Got it.
Right? This biology was still kind of a dark science of like things are just happening and we're trying to figure it out. But here, this is one of these first clues that something as fundamental as inheritance could be put into a mathematical language. And if it's being put into a mathematical language, that means there must be some like physical thingy that is going on.
That is facilitating the structure that allows you to make those effectively predictions.
Yes, exactly.
Right, about what will happen based on initial conditions.
Exactly, yeah. So it started out as a statistical abstraction that represented inheritance. And pretty soon people started asking, well, what is the thing? What is the material that is conferring this inheritance, right? So the prevailing view was that it's proteins, okay? Everyone, and it kind of makes sense. If you look back in the 1920s, 1930s, proteins have a really complicated structure. There's 20 different amino acids that we had already kind of figured out from biochemical pathways. And the fact that there's 20 different amino acids, proteins already we know have so many different functions, right? So it's kind of an obvious thing to say that, proteins are the thing that are conferring inheritance.
The idea is, proteins are where it's the workhorse.
Yes.
It's where a lot of stuff can happen.
Yeah.
It happens, not can. Literally happens.
Yes, literally, proteins are doing all these things. They're incredibly diverse.
Right.
And given the diversity of life...
It's just like, it's another thing in the toolbox.
Yes, exactly.
Which is a fair...
Totally fair.
Totally fair.
Totally fair. Right? In 1869, actually, there was this guy, Friedrich Miescher, who identified something called nuclein, which is what we now call DNA. He identified that inside the nucleus of cells, there were two different types of compounds. There were proteins, and then there was this other thing that he said was nuclein, but he was like, it's probably just like some structural thing.
He was like, "You nuclein."
Yeah, because it's in the nucleus. So I'm going to call it nuclein, and it was seen as a monotonous structural scaffold.
Okay.
Right. It's just like conferring structure into whatever is really important inside of the nucleus. Right. And then came Phoebus Levene from 1905 to 1930. He was actually at the Rockefeller Institute, which is now called the Rockefeller University. It's on the Upper East Side of Manhattan.
Yes.
Endowed by the Rockefeller Foundation.
Yes.
He discovered nucleotides. Okay. So he was going into this nuclein stuff, and he discovered that there's these things called nucleotides that really make up
That's the structure.
the structure of nuclein.
Okay.
And there's 3 different parts to a nucleotide. There's some kind of sugar, which he identified as deoxyribose. It's just a type of sugar. It's like a carbohydrate. He also identified that there's a part of the molecule that's the phosphate, right? And then if you actually look it up, there's another thing called the nitrogenous base. There's something with nitrogen. So there's 3 different parts to a nucleotide.
You have a base, you have a sugar, and you have a phosphate.
Exactly. And these are the things that make up my nuclein. Okay? That was sort of the prevailing thing. There's 4 different types of nucleotides. What he did notice was that there's 4 different types of bases in DNA. Here we're actually showing 5 because RNA, which is another type of nuclein, has another type of nucleotide. But notice the following, and this is going to be important later. With those nitrogenous bases, which is part of the building block of this nuclein, some of them have a single carbon ring. Those are called the pyrimidines.
Yes.
And then some of them have a double carbon ring. They're a little bit bigger, right? They've got a pentagon on a hexagon, whereas the other guys are just a bunch of hexagons, single hexagons.
Yes.
Right?
Yes.
And he noticed that there's these 4 different types, and he had this thing called the tetranucleotide hypothesis, which is basically that all of DNA is just basically made up of these 4 types of nucleic acids that are all common 4. And this, what you're seeing here, is the first ever proposed structure for DNA. Okay. He's like, these 4 different things, there's the A, there's the C, there's the T, and the G. That's adenine, guanine, cytosine, and thymine. Those things come together, and they create a structure called DNA, or a nuclein at that point. And this is what confers that structural scaffold into the nucleus. So this is the very, very first proposed structure of DNA. Okay, it's just like this box-like structure. With the 4 just in a little rectangle. Okay, and he's like, this is what it is.
It clearly is not what it is.
It clearly is not what it is. We know that.
We know that now.
We know that now. But back then, you can imagine like the tools that they have is not at all what the tools that we have. Right? There's no X-ray crystallography. There's no genetic sequencing. You're dealing with really crude tools compared to what we have today.
It's incredible that they were able to to get to this intuition and this directionality of what was correct. Details were a little fuzzy. But it was directionally accurate and tactically wrong.
Tactically wrong, but he had already figured out that there's 4 different types. But because he had this hypothesis that, well, they're all just like conglomerated in this square shape, there's no real way for them to be complex. They're just simple. And life is very complex.
So how do you go from the simplicity of that structure and
to all of the complicated?
And that's kind of a little
and so they're like, okay, so this is not what it is, right?
That makes sense.
Then we get to a fissure. So this is in Liverpool University.
Liverpool!
Yeah.
Shout out to all the Scousers out there. You never walk alone.
Frederick Griffith, okay? He's at the Liverpool University and at the Liverpool Medical Institution.
Okay.
And he does a few experiments with bacteria and mice. And this is what he's doing. Back then, we knew that there were 2 types of bacteria of this particular strain called S. pneumoniae. I think it's basically pneumonia. Okay? It's pneumonia that affects mice. Okay? Yes. There's 2 types of these bacteria. There's a smooth strain, which is the S, and then there's a rough strain, which is the R.
Okay.
And we know that the smooth strain kills mice,
Okay.
but the rough strain does not. Okay? You can inject the rough strain into mice, they'll live. The smooth strain into mice, they won't live.
Okay.
But here's the kicker. Here's the genius of his experiment. What Griffiths did was he said, okay, can I turn the rough strain into the smooth strain?
Can you turn water into wine? I'm kidding.
Yes. In some sense. In some sense, here's what he's doing. Like, what he can do is, so if he puts the rough and the smooth strain together, and then he puts it into mice, they die. But that makes sense because you just put the smooth strain, the virulent form, into the mice and they died.
Right.
What he does was he heat kills the smooth.
Heat kills.
Yeah. So he gets a bunch of bacteria that are really bad, that are going to kill the mice, and he heat kills them.
I don't know what that means.
Just heat it up.
He literally turned the volume to 11.
Yeah.
On the heat.
Yeah. He boiled them up.
Literally just heat.
Yeah. Just literally heated them up.
And then it inoculated kind of the fatal aspects of it.
Yes. And now when he puts that into the mice, they still live. They still live, right? Because you've killed a bacteria.
I see.
That makes sense.
That makes sense.
But here's the thing. This is the kicker.
Okay.
He takes that heat-killed strain, that little sample that he has. Let's say it's in a test tube. He's put it into a really hot water bath. It's killed all the bacteria. And he mixes that with the non-virulent type.
Mm-hmm.
And then he puts that mixture into the mice.
So he's taken the thing that's bad. He's boiled it. And now he's done a little
mixing with the stuff that was fine.
That was fine.
And then he puts that mixture into the mice, and the mice die.
So even when you heat it up, boil it all off, and still mix, there's something there. There's something there. Yes.
That's the thing.
That's the thing.
There's something physically there that has transformed the non-virulent form into the one that kills mice.
And it has nothing to do, like it's still present even after trying to have heated.
Yes.
Which the theory was that will kill any problematic.
Yeah, that's why that control was very important, right?
Interesting.
The control of let's heat up my virulent form and then put that into the mice.
Right.
They don't die. So I've killed a bacteria, but I've preserved something in that bacteria
that is continuing to propagate.
That is continuing to propagate this ability to kill mice.
Yep.
Right? This ability to be virulent. And that has gone and transformed the nice bacteria into the bad bacteria and gone and done the experiment.
The point being, there is something more fundamental than what we thought to be what is driving this process.
Yeah, it's not just the bacteria. There's something inside the bacteria that is staying put, that is going inside the non-bad bacteria, the good one,
and replicating,
making them
bad. And that's okay,
right?
Yes.
This is a very fundamental experiment. It's very simple. It's very simple, but it shows you that there's a physical element to this idea of ability.
I want to just take a quick pause to point out, like, how sometimes simple or straightforward some of this early stage science sounds like.
Yeah.
But you actually need to be able to simplify before you can complexify,
for
lack of a better way to put it.
No, totally. And with the tools that they had, this is already a huge leap, right?
Right.
This is showing that there is something there, that there's something physically there that is transforming the bacteria to become something else. Right? The bacteria is going from a non-virulent form to a virulent form.
Right.
And it's now has the ability to kill mice.
Right. Even yes.
Right?
Yes.
This is pretty crazy.
No,
already.
It is,
and this is in 1928. Right? So this is during, like, in the middle of the two wars in Liverpool that he's figured this out.
Liverpool.
Right? People are trying to figure out what is that thing.
Which makes sense. Now we've pointed out, like, there's a mystery that is clearly identifiable and replicatable.
Yeah.
What is it?
And now we get to the resolution of that with an experiment by Avery, MacLeod, and McCarty in 1944. Okay? What they do is they systematically purify whatever the thing is that is doing this transformation. And here's what they do. They say, okay, there's possibilities, right? It could be protein,
Mhm.
It could be RNA, or it could be DNA. What I'm going to do is I'm going to take that soup of heated up bad bacteria,
Right.
and I'm going to treat it with something called trypsin, which basically cuts a bunch of protein
Okay.
and inoculates all the proteins. It degrades all the proteins.
So the point is, if protein is the source, we're injecting a kill switch that
Yes.
kills the protein. And so if the protein was the source, it would not have the bacteria would not carry.
Yeah.
And
And then mice would live,
Mice would live.
right?
Yes.
They still die.
Kill switch was maybe a bad terminology to use when we
Yeah.
have double kill going on. However, the point is, trypsin killed the proteins. Proteins no longer being problematic. We put into the new mice. The new mice still die.
The new mice still die.
So it's
right?
not
So it's
protein.
not proteins.
It's not protein.
Okay. Then we do something called RNase, which we know it's something that kills RNA. It cuts RNA out.
Yeah. Okay.
Same thing.
Nothing.
Then we do something called DNase, which
Yes.
is something that cuts up DNA. We know that.
Hmm.
That's when the mice live.
Not to be confused with CRISPR, which also cuts up DNA.
Yes, I mean this was a very big precursor of this is like 100 years ago. It's a very specific CRISPR,
Understood.
right? But
Understood.
it's a scissor,
Yeah.
and what it's doing is it's cutting up DNA. Well, and you cut up the DNA that give the instructions to the bacteria to actually tell the bacteria how to infect the mice.
That's when it.
That's when we killed that ability.
Now we've found the building block.
Yes. Now we found the building block for the gene. The idea of inheritance is something that is coming from DNA.
I actually want to take a quick step back and talk about this because at this time, we did not understand how traits were passed
No.
generationally.
We
Like,
knew that they were from Mendel,
obviously.
and from all these. I mean, obviously, like I look like my parents, you look like your parents. So obviously there's something.
And then experimentally, we also proved it in a controlled context
Mm-hmm.
to know that there is this inheritance. The concept of inheritance exists.
Yes, but what it is.
What actually makes up the fabric of that?
Yeah.
It's actually a really important point to know. Like, we actually didn't know that barely 100 years ago.
It is quite incredible,
100
right?
years ago.
It is quite incredible
We
to
didn't
think
really
about.
know how I look like my dad.
Yeah. This is 1944.
That's crazy.
1944. We didn't know if it was DNA or proteins or something else.
We figured out how to split the atom and create nukes.
Yeah.
But we still didn't know why I look like my dad.
Yeah. I mean, that just goes to show you how complicated biology is, right? And how hard it is to do experiments and really figure out what the hell's going on.
We talk about this often where you talk about like, physics,
Yeah.
we've got that. But biology
Is tough, mate.
The variables are a little
Yeah, because every molecule is different. Every individual is different. In physics, we have the nice pleasure of being like that electron is the same as every other electron.
in the
Right?
universe.
In the universe, we've never found one that's different, right? So, it is an incredible experiment that they did back in 1944, right?
Got it. Okay.
At the same time in 1944, you've got Erwin Schrödinger's book called What is Life?
Mhm.
This is one of my favorite books of all time. Okay?
It's on the FFP readers bestsellers.
100%.
Okay.
It's a very short book. You can read it in about 2 hours. Honestly, it's very short. It's just a bunch of lectures that he gave at the Dublin College in Ireland, but it is one of my favorite books of all time because it really shows how a physicist would approach this problem. Okay? He was influenced by someone by a bunch of physicists at the time, like Max Delbruck, who was a physicist that had gone to University of Göttingen, which we've talked about. He was mentored by Lisa Meitner.
Yep.
And then he finally gets to Caltech, and he shows that he really starts thinking about how hereditary material must be something that is solid.
Yeah.
It must be something that can resist temperature fluctuations. And
Because of what we just talked about.
yes, and it must also be non-repetitive. What does that
Okay.
mean?
Yeah.
If it's repetitive, if it's just a bunch of repetition, you can't store a lot of information there. It's got to store a bunch of information. right?
yeah.
It's got to be like, this is how you do this, and this is how you do that. A recipe book is not repetitive.
Yeah, right.
Right?
Every page or every couple pages is a different.
Is a different recipe for all of the different things that I need in life. Right? This is it's
quite nice.
It's quite nice, but it's a fundamental thing that you can even think about for a bit and be like, yeah, of course.
Okay, that makes sense.
Whatever the thing that is giving you the information for inheritance has to be something that is not repetitive. It's something that encodes a bunch of information, like a recipe book, right? So,
because you could say you can have multiple books. Books are repetition, but within the context of a book,
yeah,
there's complexity within that that like object type.
exactly. Yeah. And one of the key things that he says, it's a postulate that he puts, Erwin Schrödinger. He says the gene is an aperiodic crystal. What does he mean by that? What he means is there's periodic crystals, right? Diamond is a periodic crystal. It's a bunch of carbon, right? You can have salt, which is sodium chloride. Not a lot of information there. There's like 2 bits of information, which is you put the sodium here and the chlorine here, and then you repeat. An aperiodic crystal is somewhere where the atomic arrangements are different, right? And that difference tells you how to do whatever inheritance thing that you're doing. Right? So he was the first person to postulate that there's some kind of hereditary code script. There's some aperiodic crystal inside of biology that is conferring information to the next generation. And this is really important because this is cited by both Watson and Crick, who are the discoverers of the double helix.
Right.
They said this was a primary motivation for seeking the genetic structure. What they wanted to do with their life was find this aperiodic crystal.
So just the idea here is that someone basically said it's a treasure map.
Yeah. There's a treasure map, and this is what it looks like.
And this is this aperiodic crystal concept with a hereditary code script, which makes you can intuit that logic.
Yeah, you can from first principles you can build it up and be like, okay, something that requires information capacity needs to be aperiodic. It needs to not be the same everywhere, right?
Right. Because your level of fidelity, if you just have 2 bits, is very low as compared to 16 or 32 or 64.
Yeah, and the order of those bits is what matters. Right? You can't just be 0 1 0 1 0 1 0 1.
Right.
It's got to be a different. And that sequence is what tells you what the information is.
Because DNA ultimately runs a variety of biological processes. And so like how are the instructions
stored?
And you need basically a system of enough different component parts or variables to store that complexity.
Yeah.
And so we have the treasure map, and then with Watson and Crick we have the treasure hunters.
Yes, that's a very good way of putting it. Yeah, they read this book and they're like, "This is a treasure that I want to find.
Let's go find the gold of whatever. What's that famous place where they had all the gold? The ancient place that back in the day.
Yeah, I know what you're saying. Okay. I saw National Treasure 2, one of my favorite movies. It's the City of Gold, right?
It's the City of Gold. Yeah. The City of Gold. They were looking because, frankly, with the things we live, we'll get there. City of Gold.
So they're trying to find this thing. They read this book.
Yes.
It's an incredibly influential book for them.
Yes.
And they're like, we want to go find what this aperiodic crystal is and what does this crystal look like.
Right.
Okay. This is around 1944. So at this point, people are already trying to justify that DNA is really the thing that carries this genetic information.
Right.
Okay.
Right.
Erwin Chargaff at the University of Columbia in Manhattan. I should say Columbia University. He uses this technique of partition chromatography to look at what is the base composition of this DNA across species. Okay. Because, back then when Levene was trying to look at the base composition, he just said everything's the same, right? There's an A, a C, a T, and a G, and they all just come together and it's all equal parts. He didn't have the resolution to figure out if there was more A or more T and so on and so forth, right? Mm-hmm. Chargaff uses this new method called partition chromatography to quantify how much A is there in a species, how much T is there, how much G, and how much C.
As an analogy, it's like you can say, "Oh, it's all soda," but it's like, is it Coke? Is it Sprite?
Yeah.
Is it Fanta?
Yeah, and are they all in equal proportions?
Right? Like, is it Coke Zero?
Yeah.
Right?
Yeah.
Or Diet Coke? What's the sugar volume? I'm trying to identify, like, create an.
Well, there's only 4 different types of sodas, right?
4. So it's, so Coke, Sprite, Fanta, grape soda.
Yeah,
we'll say those 4.
And those are the 4. But Levene has said that they're all equal, and they're just creating the scaffold, right?
Okay.
And Chargaff went out to say, okay, is this actually true? Okay. And he actually dismantled that hypothesis by showing that there's species-specific variation, meaning like the amount of A's and T's that I have is different from the amount that the cow has, is different from the amount that a pig has from a bird, and so on and so forth.
That makes sense.
He came up with a rule that said there's actually a pairing ratio.
Okay.
The A and the T are always the same amount.
Interesting.
And the G and the C are always the same amount.
Okay.
And if you look at that, this is an expert from his paper. The source is actually, this is for humans. It actually comes from human semen.
We love it.
Yeah. I mean, these guys, when you're doing science at the cutting edge, you got to get down into the weeds.
You got to go to the source.
Yeah. And the advantage of human semen is like most of the stuff is DNA.
I get it. It's a good source.
Like with normal cells, there's so many proteins that like it's gonna cloud whatever
it's a second
chromatography that you're
source.
doing. With human semen, the sperm cell is like mostly just packaged DNA and a tail.
So it's pure. It's pure.
It's like it's
sigma. It's pure alpha. Pure
alpha.
Alpha.
Exactly. Like the most of the stuff that's in there is like your DNA.
It's what you're looking for.
It's what you're looking for. So he used that and he showed that. With humans the A and T's were about 35%, and the C and G's were the rest. And then when he looked at, let's say, cows, he found a different ratio, and with pigs, he found a different ratio, but the ratios always came in these pairs.
So the point is, A and T's like to hang out, and C's and G's like to hang out.
Yes,
and that's true across species.
The numbers could be different.
The proportion of it is a little different.
Yeah,
but in everything, the A and T's are hanging out, and the C's and G's are hanging out. Okay,
exactly.
Which is like another level of fidelity that we're going down.
Yes. No, so we're getting clues into what DNA is, right? We're getting slowly, we're honing down into this. These are called Chargaff's rules, which is the A equals the T, the C equals the G, and they all add up to 100%.
Everything we have today is building off of the shoulders of the people who came before, who asked maybe simpler questions.
Yeah.
Figured out the simpler question that allowed us to ask now more complex questions.
Yes, exactly.
And we're kind of going through an example of that journey.
Yeah, and I that's why I love this story, is like it's really building up this, crown jewel.
Right.
It's not just randomly these guys came up with it.
They didn't walk into a cave and find it.
Yeah.
They had to be crafted.
There were a bunch of people. There was an entire community trying to figure this out, right? And maybe these guys just got lucky.
Right.
Right.
Look, that happens.
It happens. And now, after all that, we get to the setting of the story.
After all that,
1951, England.
Oh, so what's great is we were in Liverpool. Speaking Scouse,
we were in Liverpool. We were in Columbia. We were in Rockefeller Institute.
Yes.
In Manhattan. And now we're finally getting to the room where it happens.
The room where it happens.
There's 2 rooms where it happens, it turns out.
Okay.
In the Cavendish lab, which is the physics department of Cambridge University.
Yeah.
And King's College London.
I'm feeling a little Cavendish right now.
What?
I don't know.
Yeah. Yeah, but those are the 2 rooms where it happens, okay? The Cavendish lab, which is a storied lab.
Yep.
It's where all sorts of stuff have happened, right? J.J. Thomson discovered the electron. This is the, crucible of experimental physics before America took over post-war. Okay? And this is actually one of the last big things that they did.
Tough.
But it's good. They had a good run while they were at it.
And look, we appreciate
Oh yeah,
the work that was done.
incredible work. And one day when this podcast is big, I want to go there and record.
We're gonna do a lot. In the room where it happens.
The room where it happens, because they still have the old Cavendish lab there. They have a new building now in Cambridge that is off campus that has all of the big facilities, obviously. And Cavendish lab in Cambridge, don't get me wrong. Okay, Cavendish lab in Cambridge is still one of the foremost institutions for experimental physics research, right? But the old building, which is in the center of town, in like the old Gothic architecture, I mean, you can't beat it. You can't beat it. It's kind of like the Frisk Campus Center
I was gonna say,
at Princeton, right?
Yeah.
Frisk Campus Center, there it says Palmer Physics Lab, and you've got these statues of Benjamin Franklin and Joseph Henry, which are some of the founders of American physics. You can't beat the building where Einstein had an office, right? You know, now we've got Jadwin Hall, which is, might I just say, the most depressing piece of architecture on Princeton campus, built in the 1960s, brutalist architecture. I saw The Brutalist. It's a fine movie. The architecture is absolute trash. Can I just say?
When you look at it, because there's a lot of beautiful, amazing things to look at on Princeton's campus.
Yeah, no. And Jadwin Hall is not one of them.
It's pretty depressing.
No, it's awful. Yeah. But it does its job. It does its job, and it's still pumping out amazing research.
Look, and Nobel Prize winners.
Yeah, all the time. So anyways, let's get back to Cavendish Lab and King's College.
Yes.
These are the two rooms where it happens, okay?
Yes.
We're first actually going to run into Rosalind Franklin.
Okay.
Rosalind Franklin is this young PhD right out of Paris. She joins John Randall's group at the MRC, which is the Medical Research Council, the biophysics unit at King's College. Okay. And Randall writes to her and assigns her responsibility for the DNA project. But he does not clearly inform the guy who he already has on staff, Maurice Wilkins, that he's doing this.
Oh.
So Maurice Wilkins thinks that Rosalind Franklin is joining as kind of a lab assistant.
Here we go.
But Randall has explicitly wrote in a letter to Rosalind Franklin that no, you're going to have your own lab. She's been doing a lot of X-ray crystallography, which we're going to get into, in Paris. She's been actually investigating the structure of coal using X-rays and trying to figure out the crystalline properties of coal. There's a miscommunication there happening from the head of the lab, John Randall, and that seeds years of personal tension between Maurice Wilkins and Rosalind Franklin. These are two players at King's College in London.
Maurice is like, I'm here. Oh, you're bringing someone new? They're my executive assistant. They're going to help me take out the trash.
Yeah.
Give me coffee.
Yeah. If I'm going to be like, I need this particular scan done, they're going to do it. They're not really going to have the intellectual autonomy that is required for a true scientist.
Right. They're not going to be smart enough, and they're not going to have agency, was Maurice's viewpoint.
Yeah.
But Randall was like, no. Like, you're at
equal level.
Yeah, right.
But he didn't tell Maurice. You know? And obviously that's going to cause issues.
100%.
Right? 100%. Especially because like Rosalind Franklin is a woman. So like, it's like she's coming in at the time she's coming in. It's like, oh, this is my secretary. Right?
This is actually in the same era, maybe a little bit earlier, as Mad Men, the TV show.
No, exactly.
Just in terms for context.
It's the same dynamic, right? There's a new person that comes in. She's really good at what she does, but no one's taking her seriously, least of all Maurice Wilkins, because he didn't get the memo that this is someone that you need to take seriously as an equal. Right. So at the time, they both get into King's College. Maurice Wilkins is already there. He's established there. Rosalind Franklin is a new researcher, and they start researching DNA. They get these samples from Rudolf Signer in Austria. And this guy, Rudolf Signer, is an incredible DNA experimentalist. And what he's really good at is getting pure samples of DNA. Okay. And we've got pure samples of DNA that are coming in from this guy all the way from Austria. Right. They're originally obtained by Wilkins, but now because Rosalind Franklin is kind of this equal player, she's getting samples too. And these are very precious. because
Right.
it's really hard to extract DNA. Signer's found some secret formula to do it.
He found the Krabby Patty secret formula.
Yeah. And he's not telling anyone how he does it.
Okay.
But he's like, I'll give you my samples. Just like, cite me in your paper.
Right.
And
a little quid pro quo.
Yeah. And then we'll do it. So Rosalind Franklin, she's incredible at X-ray crystallography, which is taking pictures with X-rays.
Yes.
She builds her own camera apparatus and she starts doing humidity-controlled photographs of DNA.
Okay.
And she actually starts creating the best pictures there are ever of DNA.
Morris, eat your heart out.
Yeah. Rosalind Franklin is just incredible. Her and her PhD student, Raymond Gosling, are just incredible at taking pictures of DNA.
Ryan Gosling, no relation?
No relation. Oh, maybe. I don't know. But in this case, Raymond Gosling is learning from Rosalind Franklin.
So she has an underling, right? And they're coming in and they're just crushing.
They're just crushing.
Crushing the high quality capture using X-ray crystallography of DNA.
Yeah.
And it's just like, it's like,
yeah, and the personalities are crashing because Rosalind Franklin is very, she's a woman in science, and at the time, you got to be confrontational.
Yeah, you got to be tough.
You got to be tough. Maurice Wilkins is this really shy guy, and so even personalities are crashing.
And he always got what he wanted in his life.
Yeah, and now all of a sudden,
this tough, charismatic, attitude, he doesn't know what to do.
Exactly. Yeah. Okay. So that's the situation at King's College in London.
Understood.
Now let's go to Cambridge in the Cavendish lab.
Again, we're in 1951. These are happening simultaneously.
Simultaneously, James Watson moves there to Cavendish lab. He had just come out of a PhD in Chicago. He had done a postdoc at Copenhagen.
Yes.
And there in Cavendish lab, he meets Francis Crick. At the time, the Cavendish lab, the director was Lawrence Bragg. Lawrence Bragg is very famous for being the youngest Nobel Prize winner of all time in the sciences.
In the sciences,
right?
Yes.
He won in 1915 at the age of 25.
That's unreal.
Physics.
Right? Physics.
Yeah, this is a really, really interesting story, okay? So he won it with his dad, William Bragg. William Bragg and Lawrence Bragg won it together.
That's crazy.
Okay.
They're also the only they're the only LeBron, Bronny James.
Yeah, to win it at the same time. Niels Bohr and Niels Bohr's son also won, but at separate times. That
doesn't matter.
This is the same prize going to father and son duo.
We have a bunch of NBA players who have sons that play.
Yeah.
No one cares about that.
Yeah. But
you play
on the same team
at the same time?
Yeah. So this is William and Lawrence Bragg, right?
Cool.
They won for developing X-ray crystallography. Basically, the idea of X-ray crystallography is you've got some crystal, let's say, which is a bunch of electron density maps, right? You've got electron densities here and here and here where the atoms are. And X-ray comes in. Remember, the X-ray has a wavelength that is about the size of the atomic spacing, about 1 angstrom. So what you can do is when the X-ray interacts with that crystal, it's going to constructively interfere and destructively interfere, meaning it's going to add up and it's going to cancel out. So at certain angles, it's going to add up and you're going to get a bright spot. At other angles, it's going to cancel out and you're going to get nothing, right? By looking at the angles of where stuff adds up and where stuff cancels out, you can, by looking at the angle, what I mean is you can rotate this crystal and keep shooting X-rays.
Right. So I'm shooting a beam, a beam
of
X-rays at a crystal, and then it's going to go through that crystal. And on the other side,
I have a photographic plate.
I have something that I'm going to track, and I can move the crystal around, and I'm going to get a different map based on how, and that map allows me to actually then reconstruct
what the crystal
is, even though the map is like a 2D structure. But because I rotate and then I can reverse engineer.
Yeah. You're in technical terms, you're getting a kind of Fourier transform. In frequency space of like what frequencies at what angles are doing what. And then from that, I can then calculate. Right. And the funny thing about this, right? You think Lawrence Bragg and William Bragg, father-son duo, sounds like Nepo baby.
Yeah.
Sounds like a Nepo baby, right?
Yeah.
Lawrence Bragg, who's the son, is actually the guy who came up with this technique.
No way.
Yeah. His dad, William Bragg, was in charge of the lab at the time, and he set up the experimental apparatus. But Lawrence Bragg is the guy who came up with the mathematics and the equations for it.
That's incredible.
And when his dad, William Bragg, reported their results at the meetings and in a paper, he gives credit to his unnamed son, saying that my son came up with the equation, but the son is not a co-author on the paper. And this is something that like Lawrence Bragg had to deal with for the rest of his life. It was kind of like a trauma that he got from his dad.
No way, bro. You're telling me?
Dude, it wasn't. Yeah, dude, he's not a Nepo baby. The dad is a Nepo dad.
You know who this is? Actually, there's a viral story right now about Simon Cowell, the like judge from American Idol.
Yeah,
or whatever.
of course.
He's come out in public in the last like 2 weeks and said, my son is going to get none of my whatever $500 million empire. Zero.
Yeah.
All my money's going to go to charity.
Yeah.
And he's going to have to figure it out.
Yeah.
And it's not quite the perfect analogy, but it's like the dad basically took the son's work and was like, oh, I guess I'm the only named person.
Yeah. Oh, that's nice.
That's convenient.
Yeah. I wouldn't say that because with
That's crazy.
With the Simon thing, at least Simon's the guy who like did the thing.
He did the thing.
This is Lawrence Bragg actually came up with X-ray crystallography.
His kid's work, he put in the paper and said, my name's on it.
Yeah. Yeah. He wasn't even a co-author. They both got the Nobel Prize. But to be honest, only the son should have gotten
the Nobel Prize. The dad just had his name on the building.
Yeah.
He just had the facilities.
Yeah, so he was a Nepo dad.
I don't think you have the facilities for that, bro.
But at this point, it's like Lawrence Bragg is clearly extremely talented.
Yeah,
and at this point,
obviously.
he is the director of the Cavendish lab for good reason.
Right.
25, he wins a Nobel Prize. He's astute. And he wants, he's been using the Cavendish lab to create X-ray crystallography of a bunch of crystals. They're just getting into biological compounds, creating protein crystals and finding the structure of those. He really wants this. He really wants the DNA structure to be in England. Okay?
Okay.
In Cavendish.
Okay.
Okay?
Okay. I see what you're doing.
Right?
I see what you're doing.
And now, so he hires Francis Crick. Francis Crick is a physicist. And at the time, Francis Crick, he starts studying with a bunch of other colleagues at the time. He starts studying just X-ray crystallography in general. And he's asking, what would X-ray crystallography look like for different types of molecules?
Mm-hmm.
Specifically, he's trying to ask, what would a helical molecule look like in an X-ray photograph?
Helical meaning helix?
Helix meaning a spiral.
Okay.
A spiral molecule, I subject it to X-rays. What is it going to look like? He comes up with the Cochrane-Crick-Van theory with his two colleagues in 1952, and he publishes this paper. And what it shows is that the scattering amplitude, like where the dark spots and the light spots should be, they should come up in an X. Mm-hmm. They should have these spots, and they should have the cross shape as a signature of a helix. The angle of the cross tells you what the pitch of the angle is of that helix, right? And things like that.
So they came up with a formula for if you're looking at a helix object.
Yeah, and I have the photograph from the X-ray. What would that tell me about the structure of the helical object?
Got it. It's kind of like a dictionary or like a translation between a results list and what is the like interpretable meaning of like the raw results.
Exactly.
Okay.
So Watson at this time moves there after a PhD in Chicago, postdoc in Copenhagen, and he strikes up an immediate friendship with Francis Crick. They think very alike in terms of these scientific problems. They're very much alike, okay? So they start working on the DNA structure with whatever information that they have.
We're going to need a photo like that on Princeton's campus in black and white.
Yeah.
In the whole garb. We're going to need one of those.
Yeah, we're definitely going to need one. Yeah. So this is sort of setting up that situation, right? Cavendish
versus
Cavendish, you've got these theorists and these X-ray crystallographers that are trying to figure out what an X-ray picture would look like. And then in King's College London, they're the guys that are actually taking the photos, right?
Okay, so you have the people taking the pictures, and then you have the people creating the
theory.
framework for the interpretation of the pictures.
Yes.
And they're both kind of cooking
They're both kind of cooking,
at
right?
the same time.
Yes. And they're cooking at the same time, and they're kind of competing against each other.
Because they want to be, each one of them wants to be
each one
first.
wants to be first.
For the whole thing.
They realize what this is.
Right. Yeah.
Right? Everyone knows what this is.
The treasure map was already created.
Yes.
They know that there's a city of gold.
Yes. Yes. And they're trying to find it, right?
Okay.
Franklin and Gosling. Now we're back in King's College, London. What they figure out is there's actually 2 different types of DNA in the sample that they got. Okay. There's A DNA and B DNA, and it has to do with 2 different types of humidity. Okay. Something that's super dehydrated is called A DNA, and that's what Rosalind Franklin was really after. And then there's something that has a bunch of humidity, a bunch of water molecules there, and that's the B DNA. Okay. The way they're doing this is actually incredible. They take single bits of DNA, like strings, okay, and they pull these wires like spider webs, and they put it in front of their X-ray machine, and they take photographs with the X-ray machine, and the exposures are something like 60 hours. Jesus. They're exposing this thing because it's a tiny little thing, right? Yeah. So you need a really long time exposure to capture all of that information.
That makes sense.
Right? So there's 2 different types of DNA that they're looking at. Franklin is looking at both. She gives an internal talk at King's, okay, explaining that there's these 2 different types. The phosphate groups that I was talking about in those building blocks, those phosphates lie on the outside. And there's some kind of helical structure that they've already found because they see these X's, right? The stuff that Francis Crick was talking about.
Right.
They've already seen these X's. So they're seeing a helical form.
Okay.
Rosalind Franklin gives a seminar.
Okay.
In King's College.
Yes.
Where she's talking about her work.
Yes.
James Watson is in attendance.
Okay.
In that seminar.
Okay, so the point is we have someone from this block and someone from that block.
Yeah.
They're both trying to be the best.
Yeah.
And then so Franklin on her block is like, I'm gonna preach the gospel.
Yeah, I'm gonna do the actual experimental work.
And then Watson rolls up.
He's in the back of the seminar.
He's kind of peeping game.
Yeah.
He's like, what's going on over here?
Yeah. and he goes to the seminar.
Okay.
He's extremely excited. He comes back to Cavendish.
Yeah.
He goes to Crick, and he's like, this is all the stuff that they were talking about in the seminar. He misremembers what happened in the seminar.
He didn't have a voice memo.
No, he didn't have the chemistry background really
to
really understand what was going on and internalize. So he tells Francis Crick about all these things. They get to working.
Yeah.
They create a model, and at that time, Watson and Crick were really into making Lego models. Okay. These are like little stick figurine models of the. They're like, okay, this is what the purine looks like. It's a hexagon with these little sticks, and the stick represents a hydrogen, and the hexagon represents a carbon ring, and so on and so forth. So they start creating this model, and they think they have a model. Okay, it's a triple helix. There's 3 strands that are going out. The phosphates are on the inside because he misremembers
that it was.
But he's like really excited. He's like, oh, this might work. So the Cavendish group called the King's College group to come on down. Yeah. Get your car, come on down, check it out.
Yeah.
So Rosalind Franklin rolls up, demolishes them. Okay? In the presence of Lawrence Bragg, who's the director of Cavendish. She's like, this is wrong, this is wrong, this is wrong. This is just stupid. Right? And in Watson's garbled account, they've built this 3-stranded helix, and the data contradicts what the model they've built. Cavendish is embarrassed. Embarrassed, okay? Lawrence Bragg is like, you need to stop. Yeah. This is in 1951.
You're making us look bad?
Yeah.
Stop it.
So Lawrence Bragg is like, this is not what we do in Cavendish.
Right. One of the most legendary.
Yeah, this guy's a legend. He won the Nobel Prize at 25. He's telling you to stop. He's in charge.
Stop.
They stop. Okay? They stop. In 1951, they stop.
This is incredible.
It's actually incredible. Then, the King's College group still keeps going on. They still keep collecting data. In 1953, early 1953, they get wind of a guy named Linus Pauling.
When you say they, who?
This is both King's College
And Cavendish.
The
both-
Brits.
Okay, the Brits.
The Brits get wind of the Americans.
The Americans.
Okay? Because Linus Pauling at Caltech
Yeah.
has just discovered the structure of the alpha helix. The alpha helix is a fundamental building block of proteins. Okay? There's a helical structure that almost a lot of proteins have this helical structure that is it's called a secondary structure that is used to build the tertiary big 3D structure. Okay, you build a bunch of these alpha helixes together and you make like whatever protein you want.
It's like
but this is
it's a single Lego block that makes the thing you want to make of your Legos.
Exactly, right? And Linus Pauling comes out with this manuscript of the alpha helix. He's incredibly good at chemistry, trained under Niels Bohr.
Yeah, dangerous,
okay?
Everyone's sweating over that.
And he comes out with this. The Cavendish guys are like, wow, he just scooped us. Because they were working on the same thing. Okay. And at the same time, in 1953, Linus Pauling comes out with his version of the DNA helix. It is the same triple helix, very similar. This 3-stranded helical structure that Watson and Crick had done that was wrong.
Correct.
Right? Because he has limited data. He doesn't actually know the data.
Because he doesn't have the source.
Yeah, he hasn't been to King's College. If he was at that seminar, he's a chemist. He would have figured it out.
So the point is that at King's, they were getting source data because of their expertise in being able to do the X-ray crystallography.
Exactly.
And so they had basically the best map for where the treasure is.
Yes.
And the Watson and Crick were able to get to it because they were local.
They were local.
Down the road, whatever, and were able to see it. Now at Caltech, Pauling, he is intuiting this without having a really good map.
No. He's intuiting it from published data.
Right. From
stuff that's already out.
Right.
But this is a huge red flag for everyone in Great Britain.
They're like,
it's like, oh no, Linus Pauling is on it.
Yeah, the Americans are coming.
We got like a few months tops.
Right.
We're in the final lap of this race. Okay. No more pit stops. This is it. Whatever tires we have now, we need to get to the finish because otherwise, this Linus Pauling is like Max Verstappen. Dude.
Yeah, he's coming.
He's coming.
He's coming.
And if he figures it out, he's going to figure it out very soon. This guy has the credentials. Trained under Niels Bohr, knows quantum mechanics. Like discovered the pi bond, which is like the way in which the quantum mechanics of how atoms bond to each other. Like he's done incredible work. Okay, this guy is dangerous.
Right? If he has a little bit more information, he's gonna
do it. He's gonna do it. Okay. So Watson actually gets wind of this manuscript because Linus Pauling's son brought an early version of the manuscript to Cambridge.
I love mid 20th century.
Because there's no online, right? Like, exactly. It's not like just it goes on Nature website and then like so physically, Linus Pauling's son brought the manuscript to the Cavendish folks because he knew that they were working on it too, and they might want to see it.
Right.
Watson looks at the manuscript and he's like, okay, this guy's clearly wrong because he's doing the same mistakes that I did, but this guy's Linus Pauling. Okay, I am concerned. So he goes to King's College.
Okay.
And he tries to show the manuscript to Rosalind Franklin and Maurice Wilkins. He's like, guys, we need to set our differences aside.
This is like local. This is like in LA. It would be like two gangs in LA in different parts, right? The Blood and the Crips being like, look, we have a bigger enemy right now.
Yeah.
Can we just have moratorium for a moment?
Yeah. Like New York is coming up.
New York is coming.
Yeah.
Yeah. 50 just dropped Get Rich or Die Trying.
Yeah.
It's going to be a problem.
It's going to be,
He's hooked up with Jimmy Iovine, Eminem, and Dr. Dre.
Yeah, exactly. Okay. So this is when Watson goes to King's College.
Okay.
And he's like, look, we got a problem here.
Yeah.
He goes to Rosalind Franklin. This is where things get sketchy.
Okay.
He goes to Rosalind Franklin's office. She's not in there.
Okay.
I guess the door is open. This is where, I don't know if the door is open or not.
Okay.
Okay. He just goes in. Rosalind Franklin walks up on him in her office, looking through her stuff.
Yeah, no.
Never a good look.
No.
Okay? Never a good look. At this point, this is 1953. Rosalind Franklin has already had it with Maurice Wilkins.
Yeah.
Because of the dynamic where like they're just not getting along.
Yeah.
So Rosalind Franklin is about to leave in about 6 months to go to another university.
Right.
Okay?
Right.
And the head of the group has told Rosalind Franklin to give up all of the resources and all of the research that she's been doing on DNA to Maurice Wilkins. Hand it over, right? Because you're no longer going to be part of this group. All of that is going to go to you. Gosling, who is the PhD student, is also now going to go under Maurice Wilkins because he needs to finish his PhD.
Yes,
right?
You got to do what you got to do. Okay.
And in this whole time, Watson is in her lab, just like looking through her stuff.
Rifling through papers.
Right? I mean, the guy is desperate. I get it. But my guy.
Yeah, no. That's
like, what are you doing? Okay. Rosalind Franklin catches him. It's like, what are you doing? They have a little, tiny argument. Watson's like, well, I wanted to show you this paper from Linus Pauling. We're on the same team. Rosalind Franklin is not having any of this. Like, you were just in my office.
We're on the same
and now all
team.
of a sudden, like, we're on the same. No. Okay, get the hell out of my office. Okay? God. He's like, fine, I'll go. Down the hall, he goes to Maurice Wilkins' office. Okay. Maurice Wilkins is in his office. Yep. He goes to Maurice Wilkins. He's like, maybe I can talk to this guy. Yeah. He's like, look, this is the manuscript, Linus Pauling. He's coming at it. Wilkins has all of the data from Rosalind Franklin. Right? Because the transfer is happening.
Right.
And Wilkins shows him the famous photo 51. Okay. This is the famous photo of bDNA. This is an extremely famous photo of the X, the characteristic X that gets you the helix.
Yes.
It's so well done. This is the photo that Ryan Gosling took at...
Not Ryan.
No, not Ryan. What was his name? Raymond. Sorry,
no,
Raymond Gosling.
I got you screwed up on that.
Yeah, this is the photo. This is the photo that Raymond Gosling took. Incredible detail.
Yeah.
Right?
This is
the spots... Photo. You can tell the angle, so you can tell the pitch of the helix.
Right.
You can tell that it's a helix because of the X. The distance between the spots tells you how far apart each nucleotide is.
I want to, for people who are listening and not watching, I want to describe what we're seeing here. So there's two photos next to each other. There's sort of a black and white. If you've ever looked at photos of like people taking a picture of a petri dish, like in any science context, that's what it looks like on the left, and then on the right is a 3D recreation of the structure
of what it would look like. Yes.
And so what I want to, the point is, there's a 2D black and white image.
Yes, but it's rich in information,
and you can derive the 3-dimensional double helix from a variety of the angle and
the spacing of the dots,
all of these things in this black and white.
The fact that there's nothing in the middle means that one strand goes one way and the other strand goes the other way. So it's anti-parallel, right? There's so much information in this, and Wilkins shows Watson this photograph. Watson is immediately enamored.
So
he tries to memorize this photograph.
They were ops, kind of, because they were kind of competing with each other. And then it came to a situation where the enemy of my enemy is my friend.
Yes.
Right? Because Pauling showed up.
Yeah.
And we're like, at the end of the day, we're not going to let New York win. The Americans. Like,
we're not going to let Caltech win.
Right. So in the rapper analogy, they already won.
With Alpha Helix.
Right. So like and so now Maurice is like, okay, let me
show you what we got.
Here's what we got. She's leaving anyway.
Yeah. Yeah, exactly. Exactly. Let me show you what we got.
Okay.
Watson immediately heads back to Cambridge to tell Crick about this photo that he saw.
He saw it.
Right? At the same time, Franklin publishes an internal report, which is something that institutions do, where they talk about the research that they've done over the past year. And it's an internal Medical Research Council report and King's College report that shows shows this photograph, along with a lot of the determination that Franklin has done on the photograph, because she has the original, right? She has that original print. So she can measure the distance between those black spots and say that, okay, These things are 3.4 angstroms apart.
Mm-hmm.
Each turn of the helix is 10 nucleotides, 34 angstroms.
Mm-hmm.
And all of these little details.
Yeah.
This is a Medical Research Council report. Crick gets wind of this and gets a copy of it.
Of course he does.
Okay? All of this stuff is happening without Franklin knowing. That's the key.
Yeah.
It's not with consent.
You're right.
Informed consent.
Right.
Okay?
And especially in this world, when the stakes are as high as they are, it is a very
They're working on
meaningful-
DNA.
Yeah, this is a very
They're working
meaningful-
on the physical substrate of genetics.
I mean, obviously, they're working on a Nobel Prize level.
Everyone knows
Everyone knows
that
The
this
tr-
is what's
the
happening.
treasure map. We know.
Yeah. Right? And so Watson and Crick get to work. They have all of this information.
Right.
They go up to Bragg and they're like, look, Bragg, Linus Pauling is working on it. We have all this information. We need you to let us work on this again.
Because in 1951, he's like, stop embarrassing
Stop
us.
embarrassing us. Bragg does not like Pauling.
I love it. It's such a human story.
Yeah, it's such a human story. Bragg does not like Pauling.
And so he's on board.
Okay, so he's like, do what you got to do.
Right. Don't screw it up.
This time, do not embarrass me again. I want this for Cavendish.
Mm-hmm. Yeah.
Okay?
Yeah.
This is ours.
Yeah.
You need to do it. So, Watson and Crick go down to the machine shop. They're like, boys, we're back in business. I need you to make me little models of it so that we can tinker around.
Mm-hmm.
They get these models of nucleotides, those base pairs that I was showing you earlier, and they start trying to put them together in the way that they know now, right? Things have to be anti-parallel. The phosphates have to be on the outside. The nitrogenous bases have to be on the inside. It's still not quite working until their office mate, Jerry Donahue, looks at the bases that they're, the Lego blocks that they're using to like do this. And he's like, actually, you're doing it wrong. Given the pH of the cell and the temperature of living organisms, the hydrogen there isn't actually going to be there. It's going to be over here.
Mm-hmm.
And
Mm-hmm.
Watson's like, oh, okay. And then he goes back to the machine shop. He's like, make me new ones. And the new ones work out splendidly.
Oh my God.
They fit like puzzle blocks.
Oh my God. Like that, like singular, subtle insight.
That subtle insight from his lab mate, Jerry Donahue, to turn
Jerry!
from keto. They exist in keto and amino forms
Okay.
and not in the enol form.
Okay.
Meaning that the hydrogen was in the wrong place.
Right.
But now when the hydrogen is in the right place, the hydrogen of one is going to hydrogen bond with the nitrogen of the other or the oxygen of the other. And you're going to create these weak bonds in the middle. The other thing that was crazy was that when they had the A and the T
Mm-hmm.
and the G and the C,
Mhm.
there was structural consistency because the A and the T is pairing 2 carbon rings with 1 carbon ring and 2 carbon rings with 1 carbon ring, meaning the distance between the backbones was going to be the same,
Yeah.
right?
Yeah.
And there's you see, you've got got the C and the G.
Yes.
Those have 3 hydrogen bonds between them.
Yeah.
The A and the T have
2.
2.
Yeah.
In that case, that's a U, but same thing, right?
Yeah. Yep.
But notice that the distance between the phosphate backbone on the top and the bottom
is
are the same.
So we're looking at an image where in the middle we have on the left the G and the C with 3 hydrogen bonds in the middle, and on the right the A and the, I guess it's supposed to be a T.
Yeah, that's a U for the RNA, but it's the same
And
structure.
It's T with 2 hydrogen bonds. And the point is, when you look at it at like top to bottom, even though there's a difference in pair number, the vertical distance is identical.
Yes.
Which means it can be a structure.
Which means it can be a structure. It can be a Lego block where I can swap out A's and T's and A's and T's, but the overall structure remains the same.
Yes.
So I've just created a code
Yes.
where
Yes.
the Lego blocks themselves are different, but when you extrapolate to the larger molecule,
It fits.
it fits,
It
and they're all the
fits.
same, which means that whatever physics is happening here in this location, let's say there's more A's and less T's, in that location, there's the other way around. The physics is going to be the same. The rope is going to look the same.
Yes.
Right?
Yes. No, the
rope might be colored differently.
Right.
But the physics of the rope is going to be the same. It's the aperiodic crystal that Schrödinger was talking about.
They finally now found the missing piece for what is required to actually create the physical structure of this aperiodic crystal.
Yes.
Which initially there was like, oh, it's a triple helix. Because again, we have to remember, there's no conception
No, it's just
of this
all
structure.
in the dark.
There's no concept of what the structure is.
Yeah, they're just trying to figure out what is going on. They know that these are the building blocks, but they're like, does it go this way? Does it go this way? Are there 3? Are there 5?
And that one flip of the not enol form, but the keto and amino form.
Yeah.
That of the hydrogen.
Yeah.
Which creates the 3 and 2 pairing, which now is like, that's how everything can now click together.
Yes. And then this also explains Chargaff's rules, right?
Yes.
Because now the A's and the T's are pairing and the C and the G is pairing.
Because it's the 2 and the 3 hydrogen bonds what creates. That's the reason why they're paired in that way.
Yes. There's a physical understanding there, right? And it's an incredible story, right?
That's good.
All of this stuff coming together because they hate the Caltech guy.
Look, I just want everyone to know California is still the best.
Yeah. But they were scared. They were scared.
And they were scared for good reason because nothing's happened since. So that they were correct in their fear.
Yeah. Dude, Linus Pauling was really scaring them. So it's like, so now they finally figured this out.
Okay.
April 1953.
Yes, okay.
Is when they decide, okay, we're going to publish this.
Okay.
They have a triple publication. Okay. Watson and Crick published their very famous molecular structure of nucleic acid, a structure for deoxyribonucleic acid. It's just a 2-page paper in Nature.
No way.
It's a 2-page paper in Nature. There's no experimental findings because they didn't do any experiments.
Yeah, they just put it out.
They're like, this is the structure.
We're first.
Yeah.
Because we have to beat them.
Wilkins, Stokes, and Wilson, they provided supporting evidence of the helical structure. And then Franklin and Gosling Post had the 3rd paper in the same issue of Nature.
My God.
Back to back to back.
No way.
Yeah.
All three
back to back to back had these papers. Presented the empirical data with the famous photograph 51.
I'm going to need to get a copy of this April version.
Oh, I bet it's worth a lot of money, and I would love to have that on the wall.
So okay. So if you are currently in possession of the April 25, 1953 Nature triple publication of DNA, please DM us.
Yeah.
If you know somebody that's in possession of that copy, please DM us because it will make a great addition as one of our first pieces on the wall. We've now decided we're going to put the annals and history of science and great memorabilia on the walls behind us.
And this would be just absolutely incredible. It's a single issue of Nature that has all 3 of these publications.
That is so crazy.
Yeah.
That is so crazy.
They approached the editor of Nature and they're like, this is, we got 3 things. And they're these 3. And the editor of Nature was like yeah.
Obviously.
Yeah.
We got to put it together.
Yeah, we got to put it together. And they put it back to back.
That's incredible.
This order, though, the fact that Watson and Crick were first, it sort of cemented that priority in the narrative, right?
Yeah, right.
Because they're the guys who came up with the structure. Rosalind Franklin comes up with the empirical data that suggests that structure is true.
Right. But she didn't come up with the structure, technically.
Yeah, technically she did not come up with a structure. She was incredibly close. If you look at the laboratory notes, she has notes in her labs about how this has to be 2 strands. They're anti-parallel. All from her data because she took the data.
If Maurice didn't leak,
she could have had a little bit more time.
She would have had time, and who knows?
Yeah.
But she was... And
she was there.
She had the source.
She was there.
She was really the source.
And if you look at her notes, she didn't have the pairing, which was crucial.
That's
right?
key. That Donahue.
Yeah. And she didn't have that guy who was like, no, it's not that form, it's this other form, right? And one of the other great things about Watson and Crick's paper is they say that it doesn't escape us that the replication mechanism is now obvious because you've got a double helix, you unzip. Now there's a C here. you attach a G.
Yeah, exactly.
If there's a T, you attach an A.
Yeah.
Right? Because of the hydrogen bonding, which is unique, a 2 goes with a 2, a 3 goes with a 3, and the length constrains the spacing of the ladder constrains that an A will not go with an A. Even though an A and an A have a two hydrogen bonds, it's going to be too wide for the ladder.
That's a good point.
Right?
No, yeah, that makes
so
sense.
it's obvious now,
It's
given the
trivial.
structure, how you would replicate this information,
Right.
which was a huge deal.
Right.
Like how can you have a molecule that does both, that stores and also is able to replicate really quickly?
Right.
It's an incredible story. So now there's a shift in research. Franklin, as I was saying, she moves to Birkbeck College, and she focuses on the tobacco mosaic virus. She does incredible work on the tobacco mosaic virus with a colleague named Aaron Klug. Unfortunately, given that she was working with X-rays, it's thought that the X-ray research actually contributed to ovarian cancer that she developed later on in life. Not that much later. She died actually in 1958 at the age of 37. This is a photo of her when she was visiting California in Yosemite. And she had visited Berkeley to give some talks and UCLA to give some talks. It's really unfortunate that she died so young at the age of 37.
Yeah, that's crazy. And it was incandescently bright.
Yes, incredibly bright. And she figured out the structure of the tobacco mosaic virus. Her colleague, Aaron Klug, who was her subordinate at the time, went on to win the Nobel Prize for the tobacco mosaic virus. So very, very likely that had she lived, she would have won the Nobel Prize for that as well. 1958 is when she dies. 4 years later, the Nobel Prize is awarded to James Watson, Francis Crick, and Maurice Wilkins. Obviously, you can't give Nobel Prizes, and actually John Steinbeck won the literature prize there, so he's in the group photo as well. Obviously, you can't give the Nobel Prize for posthumously, right?
Yeah.
And Rosalind Franklin was already 4 years passed away. The problem is there is not a single mention of her in their Nobel lectures.
That's crazy.
Given photograph 51, like that is just insane.
Yeah, you guys wouldn't have been. You would have been in the dark, but for Maurice leaky ass.
Yeah, Leaky Wilkins,
giving you for his own beef that his personal tension.
Yeah,
is probably a non-zero driver.
Yeah,
in opening the books up
100%
to a local competitor.
Yeah,
right. And so you can't not.
Yeah, you cannot say anything.
Nothing?
That's insane.
Nothing is crazy.
Rosalind Franklin would have sort of gone under the radar. But nowadays, whenever we think about DNA, I'll be honest, whenever people who know the story of DNA, if they know James Watson, they know Rosalind Franklin.
I mean,
right? Because of the resurgence of her persona, and all of that happened because she was so wronged. One, she was not given due credit in the Nobel lecture.
Yep.
And then second, in 1968, James Watson publishes The Double Helix, which is this book. My dad bought this book. It's in terrible condition because he bought this in the late 90s when he first came to America. This is one of the first books that he bought. And he brought it all the way back to India, and we've brought it back when we moved back. The Double Helix was published in 1968. It's a first-hand memoir, first-person memoir about the scientific discovery. It's unusual for its time because a lot of scientists didn't really talk about the human story. But James Watson puts this out in print. He wanted to get it published in the Harvard press.
Yeah.
But Francis Crick and Wilkins were like, no, we don't want that.
Okay.
Because they read copies of it.
Yeah.
And it was a lot of self-aggrandizing.
Yeah.
The other thing is he calls Rosie by her nickname. He calls Rosalind Franklin by her nickname Rosie, which at the time was actually... No one said that to her face because she wasn't really a rosy personality, right? As I was saying, she's a tough woman.
Yeah.
She's a woman in science in a man's field. She's a tough woman. So he's calling her Rosie. He's basically saying that like Rosalind Franklin did not have the scientific expertise and the brain to figure it out on her own.
She didn't have the chutzpah.
And the men had to do it. this creates a lot of tension
That's
among all of her
crazy.
colleagues, and they're like, "What are you talking about?" And then that's what
crazy.
starts this movement to be like, "No, you're not going to get away with this.
Right.
We're going to start talking about Rosalind Franklin and her contributions." Right?
That's so crazy given the context, because like y'all were putting Legos together in the lab.
Yeah.
She was doing X-ray crystallography.
Yeah. Yeah.
And actually got the key image that created the entire unlock
Yes.
for everything downstream.
Exactly.
And to not even like just be able to
Acknowledge
that is weird, bro.
it's weird.
That's like weird.
It's really weird. It's really
weird.
weird.
It doesn't make your insight any less important.
No.
Like it didn't have to be anyway.
No. Yeah.
That's crazy.
It's weird. It's really weird. James Watson goes on to be appointed the director of the Cold Spring Harbor National Lab. This is a private, non-profit research institution. It's kind of like the Institute for Advanced Study, but for biology and chemistry and biochemistry. At the time, it was very small. James Watson really transforms this into a research environment that is unparalleled. And I will have to give him credit for that. Cold Spring Harbor Lab today is one of the foremost great world institutions when it comes to cancer research, when it comes to genetic research. They have 8 Nobel Prizes to their name. For a single institution that is not a university.
That's not a university?
It's just like an institution. It's pretty good. BioArchive, which is the biology archive, is hosted by them.
Okay.
They discovered telomeres, jumping genes, a bunch of other things. So
we just talked about telomeres.
Yes.
In one of the 2 episodes ago.
Yeah. So they've done a bunch of research, and James Watson was really one of the pioneer drivers of creating that institution. So I have to give him credit for that.
Sure.
In 1990, he actually became head of the Human Genome Project.
Yep.
Before it was taken over by Francis Collins.
Yep.
He left 2 years later because the NIH director at the time, Bernardine Healy, was proposing acquiring patents on gene sequences, and he opposed any ownership of laws of nature. So I have to give him credit for that too, because he said that genes are something that belong to humanity.
Yes.
You can't like patent something that is within me.
Yes.
That sounds completely absurd.
Yes.
So that's also really great.
Yes.
In 1994, he goes back to Cold Spring Harbor, and that's where things get unhinged. Okay. Starts getting old. Maybe he starts thinking he's invincible.
Yeah. We've seen that story before.
Yeah. So in 2000, in Berkeley, Watson gives a lecture where he suggests there's a link between skin color and sex drive.
No way.
At Berkeley.
Bro.
Like, of all institutions at Berkeley, you're going to say this shit?
For context, Berkeley is like in the Oakland area of California, which is very diverse, very black.
Yeah.
Like,
and Berkeley is extremely liberal.
And it's because they have people around anyway.
Yeah. And he hypothesizes that dark-skinned people have stronger libidos.
That's the dumbest thing.
And the lecture argues that like melanin, which is the thing that's giving us the brown and black color, that's somehow a derivative of a compound that boosts sex drive. And then, quote, he says, that's why you have Latin lovers. You've never heard of an English lover, only an English patient. Oh,
yo,
bro, what are you doing?
I cannot cringe any harder.
Okay,
that's actually that's pretty wild.
That's pretty wild, and that's not even the start, dude. Okay, so these are some other quotes from him. Whenever you interview fat people, you feel bad because you know you're not going to hire them. And then he goes on, he doubles down later, and he says that like thin people are smarter. What are you talking about?
Why is this the consistent anyway?
Yeah, earlier in the episode when I said like, this guy's controversial.
Yeah.
It's more than controversial. Yeah, no, right?
The behavior was not only problematic during the discovery.
Yeah. During the discovery he's already like
sketch. Then afterwards trying to like rewrite history. Also sketch.
Yes, also sketch.
And now you're just pointing fingers at everyone who doesn't look
now
like
he's just saying nonsense, right? And I just want to read you some of the other crazy stuff he said. People say it would be terrible if we made all girls pretty. I think it would be great. Bro, just stop talking. So at this point, Cold Spring Harbor suspends any affiliation to him.
Wait.
There's he doesn't even leave the Indians alone.
Yeah,
so this is what he said about the Indians.
There's one more.
Yeah,
quote
there's
here.
one more. I got to keep going. He doesn't leave the Indians alone.
Yeah.
He's like, Indians are servile because of selection under caste endogamy. Caste endogamy meaning castes, marry within castes in the Hindu tradition.
Yeah.
And because of that, we're servile. Nothing to do with maybe the 200 years of colonialism that was subject to us by the British.
We love to never touch on that.
Yeah.
It doesn't
And
matter.
the other thing is, look, with the caste system, it's like he's only basically interacted with the top tier of castes, probably, because the way the caste system works is the lower castes haven't really had the kinds of opportunities that the higher castes have because of the messed up nature of the caste system.
Correct.
So the stereotype the argument doesn't even make sense.
100%.
Because he's interacting with the higher, it's like at least be scientific in your bigotry.
Africans are less intelligent than Westerners. Watson said his intention was to promote science, not racism. But some UK venues canceled his appearances.
Yeah, he was going on a book tour, and the UK canceled his appearances. In 2007, the Cold Spring Harbor Laboratory suspended any affiliation,
even though that he was the father.
He basically created, but I mean, he's doubling down on this stuff. He's not even apologizing. He's just saying that no, this is how it is. And there's this YouTube page called Web of Stories. Okay. It's one of my favorite YouTube channels.
Big plug for Web of Stories.
Yeah, it really is, because what they do is they have these long-winded interviews with the titans of science. So they have James Watson, but they also have Freeman Dyson, for example, Hans Bethe. They also have Edward Teller, and it's just stream of consciousness about their life. Yeah. Okay, the ones from Hans Bethe and Edward Teller I really enjoy. They talk about their time in Manhattan Project and all of the physics that they did. They talk about their experience with Niels Bohr and things like that. Right. So they also did James Watson because he is a titan of science. Let's be honest. The 20th century, he discovered DNA, the structure of DNA. He got there in weird ways, but at the end of the day, he's the one who got there first.
He won
that race.
Max Verstappen, he might be a little bit of a dirty race, but he wins.
Yeah, he wins. And so there was this one clip where he's asked about Rosalind Franklin. The first thing he says was, I can't give a lecture anywhere without a few questions about Rosie. It's like, because you stole her data. Okay, so he's clearly already not in a good mood because the interviewer's asking him about Rosalind Franklin, and he thinks that Rosalind Franklin doesn't deserve all of this attention. And then the most unhinged thing was like, Rosalind, she was a woman, and she was also Jewish, and they say that the woman brain does not think in 3D, and neither does the Jewish brain. The Jewish brain does not think in 3D.
Crazy, bro. What are you talking about?
He's saying this, right? And then the interviewer's like, "Oh, what about like Einstein?" Right? Like Einstein literally did like 4D mathematics. And he's just like, but
he was a man. He had a penis.
Dude, it's so insane the stuff that this guy is saying. So really, I don't really have a good appreciation for how unhinged and problematic this guy was. But I saw that video and I was like, oh my God.
Yeah.
Like, he's just saying this.
Like, it's everyone, it's fair game on everybody. If you're a woman, if you're a person of color,
if you're
Jewish,
Indian,
like it doesn't matter. Like, unless you're Watson, you ain't shit.
Dude, it was insane. I was watching these videos just going like, this is the guy who discovered the double helix and like that we celebrate. And I don't even know what to think, right? Because clearly he's a great scientist, and he's a great thinker, and he's a great manager of science. But man, oh man,
terrible human being.
I gotta say,
that's
one of the worst.
Yeah, I mean, there was such a great opportunity to actually uplift Rosalind in that context.
Yeah,
like and have this like the UK is better than you. Like, you could have had a whole like narrative and story around.
There could have been so many other ways to go about
and
handling that situation.
And what's interesting is when we were growing up and we heard the story about Watson and Crick, none of this stuff was really kind of like.
No, dude,
talked about.
I read the double helix. I read this version, right? And it just shows that he's like this brilliant guy. Everyone else around him is dumb.
Yeah. And like you take it at face value.
Yeah.
Which is why if you're gonna do anything, make sure you write your own biography, autobiography, because the world will make the story up of your life. You either die a hero or you live long enough to see yourself become the villain.
Yeah.
Watson lived long enough to see himself become the villain
but it's out of his own making.
Right.
He didn't have to say any of these things.
No,
he could have just been a quiet old man.
The thing is, he didn't have to say any
No,
of it.
none of this.
This was so cool though, because like again, I know we kind of talk about it. If you've reached this part of the episode, in the comments, I want you to put Watson is not the guy.
Yeah, that's a good one.
Watson is not the guy. If that's how we'll know that people like really enjoyed this journey. I really enjoyed this journey.
Yeah.
It's because DNA is one of these things that again we learn about in school. We talk about it's Watson and Crick, and it's like that's the only part of it. Understanding this like Cavendish versus King's
versus
Caltech sort of like energy, the starts and stops, the actual fundamental science that led to the like. How do we know?
Yeah.
How do we how did we get there? It was a triple helix at first. Crazy. Yeah. Obviously dumb.
Yeah. Now we're thinking about it. It's like
triple helix, you fucking idiot. Like, asshole. But really, like, and why, like, from an image, from a 2D image of X-ray crystallography of like what the double helix, being able to then extrapolate to the structure.
Yeah. And
shout out to Donahue, who no one ever talks about.
No.
But it's kind of like the last
little peg,
peg in the structure to like get to the point of insight. I think one of the most important things that I know as someone who works in tech startups and has been building teams for a long time is it's never about any individual. It is always a team effort, whether that's one person gives 98% and another person gives 2%. Or 50-50, or there's 100 people, or there's 1,000 people. We are not sufficiently all-knowing as individuals.
Yeah.
And it requires collaboration.
Yeah.
It requires teamwork and just dogged commitment and unwillingness to give up.
Yeah.
To get to like these places. Like this is such a cool...
It's such a cool story.
It's such a cool story. And because of and again, science
and these characters are so, it's like Game of Thronesy.
Yes.
You know?
It is, all science is a human story.
Yeah.
And we've talked about this before on a pod where it's like, even when you do a research paper, you're creating a narrative because raw data does not have a through line that is digestible without framing and reference and context. An unbelievable story. Unfortunately, James Watson, again, as we mentioned at the beginning of the story, has recently passed away at age 97. Clearly, karma does not exist.
Yeah.
Because, that's a whole thing.
Yeah, he lived till 97. Rosalind Franklin only lived till 38.
38. It's like, how does that work? However, one of our best deep dives.
Yeah, I really enjoyed doing the research for this.
This was a good one. We're in December now. It's the holidays.
Yes.
As we get to the last episodes of season 1, which will round us out for 2025, we are likely to do some probably best of episodes. Maybe we'll do a little bit of a twist. Had this idea of analyzing and breaking down the science of movies and TV. Famous scenes we all know. Like when Dr. Ian Malcolm talks about chaos theory.
Yeah.
And the amphibian DNA, and you can just take some frog and mix it with so the amber, or we're not going to do Interstellar. Everyone's already done that. But we're going to try to do some concepts as we get into the holidays and it gets a little bit difficult for us to shoot to keep you guys tided over with the best content. You guys are the best audience. We're so grateful. My name is Lester Nare, as always, joined by our co-host and resident PhD and the better brown person than Chamath, Krishna Choudhary. This is From First Principles. We'll see you guys next week.
Peace.